Welcome everyone to this week's edition of the Extracellular Vesicle Club. This is an event that is endorsed by the International Society for EVs. And this week we have a paper that has created quite a bit of buzz. And so I've seen this presented at a meeting in the fall in the Northern Hemisphere last year. And it's quite fascinating. Somebody on social media this week described it as a democratization of single vesicle analysis. And indeed, there's many groups are working on trying to characterize single EVs and other small biological particles, and doing that in a multiplex fashion, really understanding the phenotype of these EVs. Numerous companies have come up with technologies that may be very expensive, maybe not so expensive. But here today, we're going to hear about a technology that will allow us to use laboratory equipment, standard laboratory equipment, to analyze our EVs. Along with a new plugin that will help us to sort through the data. So I'm going to hand this over now to Nicole Meisner-Kober, who's joining us from Salzburg, Austria. So Nicole, welcome. And I'd like for you to have the chance here to talk a little bit about what your group is doing and then to hand it over to our speaker today, Melanie Schürz. Go ahead,
Thanks.
Nicole.
Yeah, thanks so much, Ken. And it was a very nice introduction, and I'm very glad to hear about some of the feedback that has already been around about this work, because that was really our intention to share something of value to the community, but driven by our own needs. So what I'd like to do, and Melanie, perhaps you can advance one slide, is to just put things a little bit in context into what my lab is doing and why we are doing what we are doing. And in that context, I'd like to take even one further step back, because before my academic life, and I moved into academia only a few years ago, in fact, actually, I was working for many years in preclinical drug discovery in the pharmaceutical industry, with a particular focus on drugging RNA biology, both with small molecules as well as RNA therapeutics. So I'd like to share with you two learnings that we've made working on therapeutic siRNAs that have been, to me, quite fundamental and decisive, and also and still very valid in relation to what my lab is focusing on and interested in today. So back in 2007, when I was with Novartis started a program working on therapeutic siRNAs through a collaboration with Alnylam, which many of you might be aware of. And what already back then, sort of our mantra is that what we're trying to do is to drug the undruggable by making the undrugs drugs, because RNA and naked chemically unmodified RNA is really very non-drug-like molecule. You can circumvent the poor pharmacology to a certain extent with chemical modification, but really what was very clear back then already is that it needs to be facilitated for many applications with the help of formulations. So as early as 2007-8, we were already working on early generations of lipid nanoparticles, and already back then it was quite clear that these formulations work. They allowed to result in silencing in various tissues, obviously mostly in the liver in vivo, but it was always clear that's all about the therapeutic index and about adverse effects that's related to the cationic lipids within the lipid nanoparticles. So one of the efforts that we had in the group, and perhaps you can advance one further, Melanie, was really to identify rate limiting steps for lipid nanoparticles, both in delivery into the tissues and across biological barriers, but also what's happening within the cell, actually, once the siRNA is delivered into the cell. And a postdoc in my lab back then, Lukas Stalder, was taking on this project and trying to understand where within the cell a rate limiting step might be upon LNP delivery. And it turned out that back then it was also not quite clear where within the cell RNA silencing is happening. So in his work, he was really able to show that this is happening and that RNA silencing is nucleated at the cytosolic surface of the rough ER membrane, which by now I think has been documented in many systems and also holds true for microRNAs. And what was really remarkable is that trafficking to the ER, to the site of action, was a major rate limiting step for the LNPs that we worked on back then. And that it really less than 1 in 1,000 and 1 in 10,000 molecules making it into the cell actually was delivered actively and functionally into risk. And that was to a large extent due to the fact that within the cell, the LNPs didn't deliver it effectively to the site of action. So that's a long story short, but that was one of the learnings that we made. And in parallel, and we can advance one more, we also started to become interested in what we thought exosomes at the time. Today, we obviously know we're a little naive to think we were only working with exosomes, but rather with a range of extracellular vesicles. But what we were interested in is to not necessarily use them as delivery vehicles back then, but rather to understand how they function, how they interact with recipient cells, in the hope that we might be able to identify minimal components that we could retro-engineer into the lipid nanoparticles. And while we never managed to really identify such minimal components, and I think that would still be something that's would be, I think, an amazing discovery, we really were fascinated by the work that a graduate student, Wolf Reusemann, in the lab was doing, tracking the fate of extracellular vesicles, or actually CD63 GFP positive single particles, into the cell, where he was able to show that, at least in the systems that we studied, we found them recruited actively by cellular filopodia, and instead of fusing with the plasma membrane, which we were never able to document in our hands, that they were taken up as intact single vesicles into endosomes, which were targeted specifically to the endoplasmatic reticulum. So the circle somehow closed with the limitation in lipid nanoparticle delivery. And so Melanie, if you can advance once more. So basically what the conclusion from this work was that uptake of extracellular vesicles, in particular CD63 GFP positive vesicles, is fundamentally different from anything we had seen with synthetic lipid nanoparticles, with a very specific and effective targeting to the endoplasmatic reticulum. And already then we realized that tracking single vesicles, and like many others in the field, I think at the time realized tracking single vesicles is something that's extremely powerful to unravel the biology and to really understand the fate. So drug delivery through extracellular vesicles clearly has an enormous potential, because we're dealing with something that's fundamentally different from anything synthetic and more reminiscent of highly infectious pathogens. So Melanie, if you can advance once more, perhaps. So actually in 2018, when I left the pharmaceutical industry to start Manu Lab at the University of Salzburg, that was influenced, or the focus of the lab has been influenced to a large extent of learnings like these that I shared with you from our previous work. And so what we're interested today is really to bring together the biology of extracellular vesicles, LMPs, and viruses to come up with novel and superior drug delivery strategies to enable RNA therapeutics to really endorse their full potential and make their full potential hit the pharmaceutical pipeline. We have a particular interest in food-derived EVs and oral applications. And along that, obviously, in a lot of the work that we're doing, we're interested to not only quantify and characterize EVs at the single vesicle level, but also track them in different models such as cells, but also tissue, ideally at the single cell, single vesicle level. So in that context, that made us come up with strategies that don't require necessarily buying dedicated instrumentation, but really use ideally standard laboratory equipment and software that we can have easy access to adapt it to our own needs to track and characterize single EVs. And that's what Melanie is going to talk to you about, because we realized that might be tools not only valuable for us and useful for us, but really for the community. So Melanie.
Thanks a lot. Also, I will take over here. So as you already heard, my name is Melanie, and I joined Nicole's lab 3 years ago approximately. And when I started there, we were really focusing or investing a lot of time to get a standard procedure for single vesicle imaging. This was work which I primarily did with my colleague Maria Jaritsch. And what we realized when we started this was that it was not so easy as thought initially that we also quantify these EVs. And we especially if you have more than one image and you want to quantify really large data sets, we saw that it takes a lot of time. So we asked for help on that, and we were lucky because my partner Joachim Danmayr is a software engineer. So he helped us with with writing small macros and small scripts to make these characterization or these analysis of the AVs more automatic. And within the last 3 years, these small macros evolved into this EVAnalyzer plugin, which is now capable of doing a lot of different things that are interesting for the EV field. And so, as we said, that we thought it might be also important for the community, we wrote this up to a manuscript, and within the manuscript you find the plugin described, but also you find a lot of application examples how this plugin can be used in a daily EV research lab. So I think for this community, it's quite clear, and we also already heard that it's quite important to go into single vesicle characterization, because there is an inherent heterogeneity of vesicles, and also we have a lot of different isolation methods in the field. And as Ken also already said, there are many companies that came up with different technologies to help with these needs. And one of these is, for example, image flow cytometry, or some companies use imaging after capture on a chip. And we know all that these companies have a great value for the EV field to characterize these EVs. But it's limited because it always requires dedicated instrumentations or expensive chips and consumables, and also proprietary software. So so it's really limited to labs also that are highly specialized on the EV research. And so driven by our own need, as Nicole already said, and we came up with a protocol for single vesicle imaging. And single vesicle imaging basically requires 3 steps. One step is the immobilization of the EVs on the slide, where we have an SOP developed, which is cheap, fast, and robust. And secondly, we need to get the EVs imaged, which of course works on super resolution microscopy and also on confocal, but we show that it also works very well with standard wide field microscopes. And then we also have the EVAnalyzer, which is this plugin for ImageJ, which is open access and open source. And it's really optimized for images from the EV research, and also it's optimized to have a high throughput. Basically, the plugin consists of 4 different packages. So first of all, we have different filters which are used from Fiji mostly, and they are just a bit altered because we wanted the analysis process to be more fast. And then we created different pipelines which were created to really get single vesicle quantification and also single cell single vesicle quantification. Then we have the output and the graphical user interface. The graphical user interface looks like this. So it's really designed to be easy to use, which we already tested on some students. And what you see here is all the infos you have for the individual lines. And then here you can put all settings for the individual channels that you want to characterize. In general, we have 5 different functions or 5 different pipelines that we can use to analyze images from the EV research. 2 pipelines are made for single vesicle quantification. So for this, we have either EV count. So here you can use single or multi-channel images. And what's happening in this pipeline is that we have an input image. We do some pre-processing, like, for example, we use a rolling ball to subtract background. And then we do thresholding, and the EVAnalyzer then counts the events that are found, or the particles that are found on this image. And Additionally, for some applications, it's also recommended to add some calibration beads, and like, for example, tetraspec beads. And these calibration beads or tetraspec beads can be easily automatically also removed by the EVAnalyzer. Secondly, we also have a EV colocalization pipeline. So this is, of course, only possible with multi-channel images. So we have the same pre-processing steps and thresholding steps, and then with the different channels, microscope channels, we the EVAnalyzer makes an intersection and calculates how many of these detected spots are colocalizing. Generally, the EVAnalyzer by default recognizes everything that bit colocalizing as colocalizing. So if we have two particles like that, it will count it as colocalizing if they are only a bit colocalizing. So as we saw with our data, with also processing a lot of data, that we also see random overlap, especially if there is barely any colocalization error, then we mostly see with true colloc that they are really more than 90% overlapping. So what we did was we also included a minimal colloc factor, so you can decide yourself if you how much percent of the spots have to be colocalizing to be counted as a true colocalization. In this current version, we can until now colocalize up to 5 channels. Then next, we also have some pipelines for single cell and single vesicle quantification. So this is used for cell uptake. So if you want to know how many EVs are taken up into the cells, and here we can either calculate the EVs per cell area also, or we can also go into really calculating the EVs per single cell. But I will go a bit more into detail on this a bit later. Generally, the EVAnalyzer is programmed to be really fast, because we always look at a large data set, and so it's programmed to do high throughput batch processing, and we tested it for more than 10,000 images per run, and depending on the image size, it's mostly faster than 1 second per image. What is also nice is that we included some analyzer scaling, so you can gate your output by parameters like size, circularity, or color area. And we also included some automated image pre-processing steps, like, for example, set projection can be done before the image is analyzed, or edge detection or image cropping. So this further minimizes the manual working time. And we also included a preview function, which is really helpful if you want to set the right thresholds. It opens the first image in your folder, and you can see how it would look like with the threshold that you set at the moment. And we also included some new features since the publication. One is that we can use now also multiple CPU cores. So what you see here is the EVAnalyzer is automatically telling you how many CPU cores your computer has, and then you can tell it how many it should use to do the analysis. For example, if I use 6 CPU cores, then 5 images are processed in parallel. So you get the analysis done 5 times faster even. And we also included a test run. So if you have your parameter set, you can test this on 2-3 images, and you can easily select these images in this test run. During the analysis, the EVAnalyzer automatically creates these control images. And here, for example, you see a control image or control image from an EV count from 2 channels, where we use the Lie auto threshold, and you can then later have a look if what was counted as an EV, and how the EVAnalyzer gets its result. Here you see the image of a cell uptake, and this is how it looks after the processing steps or after the pipelines done by EVAnalyzer. Additionally, it also generates an Excel output, which is done when we have less than 1,000 images. With more than 1,000 images, it generates a CSV output, and it also stores a JSON file. So in these JSON files, we have all the settings stored, so you can easily reload the settings for different data sets. When we look a bit more into detail with on the output Excel file, we see we first of all get an overview sheet. So this is here in the top. And what we get in this overview sheet is for each image that was analyzed, one line is created. And for each of these lines, you get all the informations for your EVs. In these examples, we have GFP labeled EVs and Sci3 labeled EVs, and we look at the colocalization of these EVs. So we get all the information for the EVs with the GFP, area size, intensity, circularity. This is always the mean circularity of all EVs in your image. And then you get the number of particles that are counted. So valid is the number of particles that fulfill your criteria. Invalid is the ones that do not fulfill your criteria. If we look more into detail on the detail sheet that you also get, you see that for each spot or for each detected vesicle we get all of these informations. And with validity, you can have a look. For example, here, validity 4, we know that these particles were too small, so they are filtered out and you find them in the invalid column. That's how you see how much of the vesicles are filtered out by your gatings. And you also get information about the colloc factor here, so how much area of your EVs are colocalizing. All of this information can also be easily found in the handbook. So the handbook is attached to the supplement info of the paper. But as we always have new features, the new version of the handbook can always be downloaded by GitHub and on Joachim Danmayr's page. And what you find in the handbook is general system requirements. You find information about how much storage space you need for these control images. You find how you install it, and also how it's generally started. And you get information about all the gatings and settings you can do. You get all the information about the output, what it exactly means. And also at last, we also included some examples how you can put your settings so you can do an EV counting or EV colocalization. So just to have an easy start. Next, I want to give you some application examples for the EVAnalyzer. So we are using it really on a daily basis, and there are a lot of application examples in the manuscript. I will only give you some examples because it would take too much time to really get into each example that we have given. So first of all, I want to start with one really important example, which is immune labeling of extracellular vesicles. And for this, we first optimized immune labeling protocol on extracellular vesicles, because this was something that, there are basic immune labeling protocols, and which are basically these 5 steps, but we had to still really optimize it for extracellular vesicles. And so what we did is first we looked into immobilization of EVs on different surfaces, and what we used was normal microscope slides. We used IBD slides or these Evotec 24-well slides. We also used different coatings like collagen or poly-D-lysine for coating, and we used 2 different methods for immobilization of the EVs on the surface. One was that we let the EVs dry on the surface, and the other was that we incubated it for 30 minutes. And what we saw is that when we let the EVs dry or evaporate the liquid, we get much more EVs immobilized on the surface. But we have a higher loss of EVs when we wash these surfaces, because in these stainings we have a lot of washing steps, and they should still be on the surface after these steps. Also, what we see is that we have drying artifacts, so the EVs are very inhomogeneously distributed, and we get salt crystals. While with 30 minutes incubation, we have less EVs immobilized, but they stay after washing, and they are homogeneously distributed. And generally, we got higher quality images. So what we also saw is when we looked at the different coatings and different surfaces is that regular quartz glass slides worked really well and worked best for our purpose. So we continued with normal microscope slides and 30 minutes incubation. In order to get an area where we can put liquid, we use these stickers that are called gene frames, but you can also use a fat pen to demark an area where you can put your EVs. Next, we tested the fixation of the EVs. So we tested different fixatives to see if we get a loss of fluorophores or a loss of fluorescence signal, and this we tested on cells, and we saw that they look quite comparable. So these 3 fixatives that we looked at, but we continued with 4% formaldehyde. Then we looked at different blocking agents, and here we saw that milk powder and normal gold serum worked best because we have no unspecific binding of the secondary antibody. So in our protocol, we always use normal gold serum now. And last, we also checked different embedding medias. And what we saw is that with Vector Shield, we get a really comparable results, even if we image our slides later. While with Prolong, when we image our slides later, we see that more and more of the EVs accumulate. So this is something that why we chose that Vector Shield works best, but both work good in preventing bleaching. And what we also tried was the effect of Triton on EVs, because we wanted to permeabilize the EVs in order to also stain intravesicular proteins. So what we saw when we added Triton to the blocking buffer was that the EVs were washed away. So we do not have yet a protocol for intravesicular staining of proteins. Last, we also checked the reproducibility and of the protocol. So we had 3 different operators who made exactly the same experiment, and what we saw is that we get really comparable numbers of EVs that are immobilized to this surface, and of course also comparable percentage of colocalization. Last, at the end, we also tested we also wanted to test how trustworthy or our results we get from EVAnalyzer are. So what we did was that we asked 4 different users to analyze the same 10 images. First, ones with manual quantification. So here the users opened each image, set the threshold, and then counted these spots. And here we have the analysis done by the EVAnalyzer with the same 4 operators. And what we see is, first of all, that of course with manual quantification we take a lot of time because each image has to be opened separately, and we get a much higher image to image variation with this manual quantification and threshold setting each time newly. And these same 10 images were then also analyzed with an auto threshold. So the auto threshold can be used or uses a mathematical operation to find its own threshold. So each of these auto thresholds uses a different mathematical operation to do so. And as you see, one has to be really cautious because each of these auto thresholds, of course, finds a completely different number of vesicles. For example, when we look at mean error, we see there is a lot of background already that is found with these auto thresholds. So one really has to look at the samples and decide which auto threshold works best. What we found out is that Lie auto threshold works very well on fluorescent spots with low background, and the max entropy works well for spots with a very high background. One example how such a antibody or immune labeling can work is or can be used is to validate antibodies. So this is something that can be very useful because one never knows if the antibody really recognizes your protein. So to do so, we can transfect cells with our protein of interest. In our case, this is a CD81, which is tagged to an amyloid GFP. And these cells then produce vesicles which have the CD81 with the amyloid GFP on the vesicle. And we do the antibody staining with our CD81 antibody on these vesicles. And then we can have a look how much colocalization we see. What we see here is that we get nearly every green spot colocalizing with our anti-CD81 antibody. So this results in this yellow color, and we only have a really small portion of green-only spots, so they have a CD81 but are not recognized by the antibody. Additionally, we get a large proportion of spots that are CD81 positive, but have no GFP. This can easily be explained because there are a lot of cells that are not transfected, and they also have an inherent CD81, and so this is something that is well explainable. The same staining also works really well on conditioned medium. So as you see, we get also really nice and comparable results if we do this staining directly on conditioned medium. Something that can also be evaluated with this method, and which is especially interesting for us, was to look at species cross-reactivity of antibodies. So what we did here is to transfect cells with human tetraspanines or bovine tetraspanines. So bovine tetraspanines are especially important for us because we work a lot with bovine milk EVs. And we can see if an antibody recognizes human or bovine, or if it recognizes both. Another really important application for the field, I think, is the evaluation of EV labeling. And this is something that we did on EVs from CD63 amnion stable line, which we which was kindly provided by Samir Andalusi and Andre Görgens from the Karolinska Institute. And we stained these amnion vesicles with different labels. One was 5-NHS, one was MEMGLOW, and one was DII. And what we saw is that with MEMGLOW and 5-NHS labeling, we get almost 100% of our CD63 positive EVs labeled, while with DII, we only get a small proportion labeled, at least in our hands. So additionally to this, we can also get information on spot intensity, on circularity and size. And here one has to be cautious because size does not really refer to the real size, because we are looking at images from wide field microscopes. And so the vesicles that we detect are only detected as a point spread function. And therefore its size does not reflect the real size, but more the intensity of the vesicles. So what we did is we had a look at these different samples with the EVAnalyzer and also with the second method. So these samples were engineered with different methods. We have a Pittsburgh line and also an overexpression CD63 GFP line. And once we analyzed it with fluorescence correlation spectroscopy, and with this method, we get the molecular brightness of our EVs. So we get the mean number of fluorescent molecules per vesicle. And secondly, we analyzed it with single vesicle imaging, and we analyzed it with EVAnalyzer. And here we get spot intensities for our vesicles. And what we saw is that the brightness that we get with EVAnalyzer really well correlates with the loading densities that we measure with the FCS. So we can really correlate the intensity of the vesicles with the number of molecules on our EVs. And as with EVAnalyzer, we also get data about each single vesicles. We can also look at brightness distributions or brightnesses of two different fluorophores. One example where this can help is one experiment we also have included in the paper. So this is a simple experiment where we try to optimize transfection with CD63 emerald GFP to see where we get most of the CD63 emerald GFP positive EVs. And when we look a bit more in detail into the intensity data, we see that the vesicles that have the CD63 emerald GFP have much more signal for the antibody labeling that we did with CD63. So the ones that have the emerald GFP are have much more CD63 than the ones that are inherently produced by the non-transfected EVs, by the non-transfected cells. Sorry. Last, we also tried to use EVAnalyzer for the nanoparticle field. And this we did with some colleagues from the Mendel University, where we looked at eRNA loading in LMPs. Another application was done in cooperation with a group here in Salzburg, and where we looked at silica nanoparticles, which were labeled, or where proteins were added, and these proteins were labeled with different fluorophores. So we had a look at these different fluorophores. Sorry, my light just got, no, it's fine. Thanks, leave it. Sorry. it got a bit dark here in Salzburg. So we had silica nanoparticles, and they were incubated with these proteins, and we wanted to know if these proteins bind to the silica nanoparticles, and if there is if they inhibit each other or if they bind together. And what we saw is that we see a correlation of intensity between the Cy7 and the TAMUA. So these proteins seem to bind together. So this is something that can easily be done with the EVAnalyzer. And last, I also want to show you the pipelines that we use for quantification of EVs in cell uptakes. So for cell uptake, we need, first of all, EV detection. EV detection in this case it's more tricky than for single vesicle imaging, because in the cells we have a lot of additional background, and also the EVs cluster more. So we have different filtering steps included to really get to a single vesicle level. And secondly, we have to detect cell area, which can be done by with wide field images, but which can also be done with fluorescently labeled cells. And then if we want to separate our cells, we also have to stay in the nucleus. And with the help of the nucleus, we can form this Voronoi grid, which then separates the cell area into individual cells. With this help, we can get from this input image into this image, which is the control image that is generated by the EVAnalyzer. So with this, we get a lot of information about our EVs taken up. We get the mean fluorescence per cell area. We also get the mean number of EVs per cell. We get cell area. We get the EV number for each individual cell, so we can, for example, have a look if there is a correlation between the number of EVs taken up and cell area. Or we also get information about the brightnesses for each single EV. And we also can look if two fluorophores in the cell are colocalized. So here we have also a comparison of colocalization before the uptake and after the uptake. Something that is not included in the paper, but where we're working on, what we're working on in our lab is also to use EVAnalyzer for detection of EVs in body fluids. So here we have an example for serum, and we see there is a lot of background in serum, and it's much more tricky to really only analyze the EVs and not the background. And we also found some really nice pipelines to do so. And we already used it to look into serum pharmacokinetics. And what we see here is milk EVs that were labeled with Cy5, and we see something that also others describe, that the vesicles are cleared really fast from the blood. And we hope that these pipelines can also be applied to other samples like urine. Something that we also look into is the quantification of EVs in histological sections. So we try to dose the mice with the EVs and then make liver sections, for example. And when we look at these liver sections, we see that we can find back our EVs in the cells, and we can analyze these images by EVAnalyzer. This is an experiment where we look at liver sections with and without perfusion. And last, I want to show you some implementations that we were partly working on, which is, for example, the quantification of super resolution microscopy images, where we can also use EVAnalyzer, but this is something that can still be advanced. And we are also looking into different options for the quantification on tissue sections. So in the end, what we are aiming for is to really get on a single cell, single vesicle level. And what we also do is that we look at EV distribution across larger areas. So here you see an example for that, where we have 2 wells, and we look if different coatings lead to a homogeneous distribution of our EVs. And this is something that can be very helpful for questions of immobilization of EVs. So summed up, EVAnalyzer is an open access and open source tool, and it's directly updated via Fiji. It's released via GitHub. So here is the link to GitHub, and there you can find everything. You can find the handbook and also all the releases. And additionally, on this page, you can also add issues. So if there's something that's not working, or if there's something you think would be good to have in the plugin, you can make an issue, and Joachim then decides if he programs it. And you also find all the releases of the EVAnalyzer on GitHub. So last, I want to first of all thank you for your attention, and I also want to thank my whole group that participated in this work, and also Joachim, who did all the programming of this plugin. Thanks a lot.
Well, there was a lot to cover there, and thank you for that. We do have quite a few questions that have come in, and I see that okay. Nicole has been very busy in answering some of those, but I do believe that there are still a lot of things to discuss. So okay. Let's go to it. I think that, so not everybody sees the chat box. And of course, the folks who are watching this on YouTube later will not be able to see the chat box either. So we'll try to get through this. I see that there are some themes here. So I'm not going to call on everyone individually, but I see that Anand and Ritu and Claudia have all had questions about the staining protocols, and then also about the permeabilization protocol. So let's start with staining. Anand, do you want to just kick this off, and then Ritu, Claudia, anybody else who has questions about staining, feel free to jump in as well.
Perfect. It was a great talk, Nicole and Melanie. I really enjoyed it. So I had like a few questions. I think Nicole already answered few. Starting with, what other lipophilic stains have you tried using? Because a lot of these new model systems like invertebrates don't have good antibodies. So we have to rely on these lipophilic stains to do these things. So I think you said like MemGlow is the best compared to all the others. So I take it from that, then I'm going to try MemGlow hereafter. Another thing I want to ask is, like, you said about immobilization and you said 30 minutes, but I kind of missed out on with what did you immobilize and what was the substrate used apart from polylysine?
So we really, so what we tried was we tried different coatings on microscope slides, but we found that only plain microscope slides work really well. So if you just take your EVs, we always take the concentrations of, I think, 1 × 10⁹ measured by nanosite. So whatever that means for you then. And we just take this in PBS, add it to the slide, incubate it, remove, then we fix it, and then they stay. So they really stay doing all the washing steps.
So you don't have to coat them with any ECM or any matrix?
As I said, we tried some coatings, but as there was no more immobilization, we also tried positively charged slides, but they also didn't work better. So that's why we always use those, and it works really well throughout all these washing procedures during the immune labeling.
And I should say that by now, I mean, again, it might differ depending on the EV source. But by now, we've been doing this protocol, correct me if I'm saying something wrong, Melanie, on EVs from many different sources. So milk from various species, different cell lines, but also bacterial EVs, honey EVs, lemon EVs. So we've really done this on many different sources. What you never know, of course, is how representative is what you mobilize to what's in the solution? That's certainly an open question. But what we can say is that it's extremely reproducible. And if you change something in the purification protocol, you will see different types of particles that come up in addition. So I think this is as good as it gets at the moment. And those particles that you mobilize, they really stay throughout the staining procedure. So they survive 13 steps or so.
Perfect. Thank you.
Yeah, this is such an important point because so many of the EVs that we study, so many of the species that we study don't have these established protein markers yet that can confer the specificity that we would like to have for the phenotyping. And so the labels, the non-specific labels, thus come in very handy for those species and their EVs. So thanks, Anand, for the question. And Anand, you also, not to put you on the spot, but you also had the question about the permeabilization. And so I see that Mehdi also has a question about that, Kyriaki. So Anand, why don't you kick off too, and then we can hand this over to others who might want to ask questions about permeabilization.
Yeah, that was more like a suggestion because I tried Triton for permeabilization. It gave me like a really, I used 0.001%, and it wasn't so great. And then I moved to saponin, which kind of worked really well for me for my immuno-EMs and other things. So I just want to put it out over there. And I'm just gathering more things. If someone has any other idea, like some new detergent, happy to take a try on that also. So
okay, thanks a lot. Yeah.
So Melanie, correct me, but I think what you've tried is different ranges, or Maria has tried different ranges of Triton and Tween, but saponin I can't remember.
I think we only tried Triton actually. So I think there was not so much we did on that.
We tried also NP40S and saponin.
Okay, sorry.
And I think the question,
the issue was mostly that the EVs get lost from the surface. I think that was the problem, right?
Yeah, exactly.
Yeah, that was the main problem.
So there was also the question from Kyriaki, who asks, does your fixation method itself semi-permeabilize the EVs? So can you actually detect luminal proteins without an actual permeabilization agent?
I mean, we tried at the beginning to also label THG101, and I'm not 100% sure if our antibodies didn't work, but they work on Westerns. Or if it's due to an insufficient permeabilization. So I cannot really surely answer this question. So it didn't work for the antibodies that we have here, but yeah, I'm not 100% sure to answer this. So it could be that it works for some antibodies, but it didn't work in our hands.
I mean, luminal staining is something that, this is something I think where with additional work, there should be possibility or conditions that could be identified, but I think it just takes some extra work.
Yeah, I know luminal stuff is kind of a bugbear in my group right now. How to interpret something if you see a signal without, without that permeabilization. So, so probably a lot of optimization to perform for some of those things. Kyriaki, Mehdi, did you want to jump in with any other comments? If not,
no. Hi. Everything was covered up, I think. I was just asking because in my hands, you're right that fixed vesicles, they are kind of permeabilized. You can get signals of luminal proteins, at least in immunogold EM, by adding very low concentration of Triton helps without disrupting too much the vesicles, and you keep the shape of the vesicles. So that's why I was asking what concentration, because I was surprised that the Triton made you lose actually some of the vesicles on your slide. Yeah.
But so how do you have them immobilized, or you also have them on a slide?
Well, for immunogold, you just they're fixed and then absorbed on the grid, the carbon grid. They're nothing special to immobilize it, and then the way I do it is add a little bit of buffer with Triton on the grid for like very short time, like 5-10 minutes. And then block and do the EM. the antibody labeling, and it works. I mean, it's not like perfect, but at least I have more signal than when I don't treat with Triton. So I have more positive vesicles, let's say, for luminal proteins, the ones that I tried at least.
Maybe they adhere better to the squids than to the microscope slides.
Yeah, maybe it's different.
Yeah.
Thank you.
Good. Yeah. And I see Kyriaki has added a comment here. Thank you. We are also scratching our head about getting signal of what should be luminal, and we're wondering if that PFA might be actually permeabilizing on its own. So I think there's something interesting to be found here with that kind of aberrant, sometimes luminal staining. So let's go. I want to get to some of the software questions too that have come up, but let's first start with, or let's go back to James Drummond. James, you had a couple of technical questions here, some of which may have been answered. But so James is asking, have you tried antibody capture? James, are you there?
Sorry, yeah, I forgot to turn
hi.
I was just wondering if you had tried to capture with specific antibodies as a way to get specific exosomes, you know, at least expressing the one surface protein to stick.
Yeah, I mean, there you would have to immobilize the antibody first. So this is something I'm not 100% sure. I would hand this over to Nicole because we have another
I mean, we haven't done that, but in theory, I mean, EVAnalyzer, the tool, you can definitely use also for images that you generate on EVs that are immobilized by any other means or by any other capture method. But for us, for us, our main purpose is to get an idea of what the situation looks like within the sample. So how do these look like within the sample, and then we count them based on staining them either with an antibody or with different types of dyes. So
okay. My next question was just a simple one. What magnification are you on your microscope?
So we use either 40x or 100x.
Okay.
So both works well.
So both will work with, even though the EVs are so small.
Yeah. I mean, you sometimes take some time to find the right X set position. Sorry, but if you have that, it's working really well.
So I don't know. I mean, Melanie, didn't have so much time to also. But what we're using a lot is we're including calibration beads, tetraspec beads, to find the set plane. And that can be removed from the images automatically also. So those can be removed.
That is important if you really want to look at the intensities of the vesicles. So you don't bleach your actual vesicles, but you have an additional channel where you only have your tetraspec beads, and you focus on these. Then you really get the intensity data even better.
Yeah, that was a good addition. I like that. Thanks.
Good. Yeah, thanks for the questions, James. And now I'd like to maybe switch over a little bit. So we have a question from Clotilde Terry. She had to leave, unfortunately, but she had a very good question about co-localization, or actually interpretation of maybe two vesicles that are next to each other. So I think there's a dilution answer here, which Nicole has given. But then there also might be a software answer. So I see that our co-first author, Joachim, is also here. So Nicole, do you want to start out with how do you ensure?
I mean, this question we got a lot also when writing up the manuscript, and we asked ourselves, of course, as well. And that was experiments we did very early on, was that we combined EVs. So we took EVs from cells which either expressed CD63 mCherry or CD63 GFP, and then we did purify them either separately or we co-purified them. And then we looked at how much random overlap do we get as an indication for aggregation. And there we found that indeed you do get some aggregation if you co-purify them by ultracentrifugation, but with other milder purification methods, we found almost zero. It was very minimal colocalization of, and that was even with a wide field microscope. So we get very minimal colocalization of EVs in two colors if you combine them. If you have samples which are very prone to aggregate, and we see that actually with the silica nanoparticles, silica dioxide nanoparticles, you will find larger spots, and you will also find artificial colocalization, which you can then also discriminate with setting a higher threshold in the colocalization area.
So this is actually, we have an example for that in the manuscript, where we really have these, where we really compare this colocalization area between vesicles where we know they have both fluorophores and vesicles where we know we have one type of vesicles that have this fluorophores and the other type. And I think we had around 3% of colocalization where when we look at these separate ones, and when we add these colocalization area to over 70%, we can make this to 1% from 3 to 1. So this is, I don't know, this 1% if this is a actual colocalization or if this is somehow fusion or not, but it's really a really small portion that is then actually colocalizing, and you really see it in the colocalizing area, that if you have random overlap, because if there is real overlap, so what we saw from our data was always that we get a really high percentage of colocalization then.
Very good. All right. So let's see here. We have a few more questions about the capabilities here. Claudia, thank you so much for your contributions here. You've had a lot of good notes in the chat box. So would you like to go ahead and ask your questions, and we'll see if we can get some answers here.
Yes, of course. Thank you very much. As I said in the chat, it was a great talk. And for me, that I'm doing a lot of this image analysis by hand. It's, as you know, very tedious. So I'm definitely looking forward to give this a try. I work with bacterial EVs, which, as you also know, brings an entire, completely different spectrum of difficulties, because we don't have very defined markers as you guys have working with humans. And my questions were regarding the size, the maximum size you guys tried or tested as input files in the analyzer, because mines are roughly between 1 and 3 gigas per image. And then I also had one running question, but Joaquin answered brilliantly. I also had a running question about if this analyzer supports bioformats, and someone else also asked this question in the chat. If we can use or not, so size or
yeah. So we use bioformats to open it. So if your whatever image format you have is possible to open with bioformats, it should also be possible for the EVAnalyzer to open. And I'm really not sure about how big the images can be, but we sometimes have 7 channels and they are really big, and also with set stacks where we do set projection first. So I don't know if there is a limit, because we have these really huge images sometimes which didn't work differently. I mean, they take a bit longer to process, but that's it. So I think there is not really a limitation, but I'm not 100% sure if there's a limitation, but it works with 7 channels, and I think 7 set stacks, and yeah.
That's great. Yes,
I mean,
that's
also
very helpful.
maybe Anna, I don't know whether Anna is in the chat, but so Anna in the group is doing a lot with histological sections, where she's also stitching the images first, and these images can be really big. And so she can use EVAnalyzer. So if there is a limit, we haven't yet hit it.
Great, thank you very much. And then I had one last question, also already answered by Joaquin, about the denoising algorithm that is running behind, because you were mentioning all this thresholding, and of course that we need to try a little bit and find the best things. And then I think I missed the denoising, but I guess it's inside there. So it's,
I think, so you find all the steps in the supplement info also from the paper, but so we use a rolling ball for the single vesicle imaging, and what we do for the in-cell imaging is that we kind of, so the problem with the in-cell vesicle quantification is that you get a lot of clustering around the nucleus, and so this area around the nucleus is really bright, and if you have a vesicle on the side of the image, it's not so bright. So when you only put a threshold, you get one big area in the middle, and you lose all the vesicles on the side. So what we did is, or what Joachim came up with, was the idea to first blur the image to really get a general brightness, let's say, and then subtract this brightness. And then you get really just these single spots, and it's then really easy to, or it works really well to really get these single spots that you see with the eye. But as the background is so bright already, you cannot really use a threshold. So this is something that we use then for this pipeline.
Great, thank you very much.
Another good question from Theodora Katsila. Theodora, are you still there?
Yeah, hi all. Apologies, I don't have my image and you cannot look at me. We have snow in Athens, Greece, and everything is paralyzed as we are a summer country. We don't know how to deal with snow. So sorry about that. Great talk, great paper. I have been reading your work a lot. Huge congrats. I had a question, and I feel Joaquin truly replied, but I would like to have your feedback as well. So we do understand the heterogeneity of exosomes or EVs, sorry, and there is a lot to be done depending on different biofluids, etc. I saw the question and the answer regarding the serum. I was wondering if you would be interested in applications regarding other biofluids like plasma with like a hole of a pain due to platelets and artifacts from such sources.
Yeah, I mean, we only looked until now into serum. I have no idea with other biofluids how much background there is. If maybe it's better than with serum. I don't know.
I think serum is already quite bad.
I think, yeah.
I would assume urine should be easier in terms of background fluorescence and artifacts.
I don't know how easy.
So we just got to try. Yeah, we just got to try. So if you have some,
it is in urine. But we would be really interested to also look into this and to see if we can analyze it with it, because if it works for serum, I think, I mean, if we can find a way how to put together different filters to get it, we can put it in a pipeline and add it easily. And I think the tricky part is to find the right pipelines that is really suitable for different kinds of images and different kinds of background. So maybe we have to adjust the pipeline we use for serum a bit for urine or for other body fluids. But I mean, I think
so Theodora, if you're interested to follow up, I mean, just feel free to reach out, and then we can see whether we could follow up on this together offline.
Yeah, great, Nicole. Yeah, that would be great. Thank you both.
Thanks, and stay safe in the snow there, Theodora. Thank you. So we are coming to the end here. I think related to this question about background, there's a question from Winfried Neuhaus who asks, do you know whether nanoplastics like polystyrene beads or other materials might also stick? I guess the question is here, is this something that would be applicable to other nanoparticles?
So what we saw is, first of all, with lipid nanoparticles, is that it works well for those, but they are really they're a bit trickier to handle than EVs, because they should not run dry, because otherwise you don't get them nicely imaged anymore, which works well for EVs. So you can also have them dried if not too long, and everything still works, which does not account for lipid nanoparticles. With beads, we also only tried silicon beads, and those clustered a lot, and the immobilization was quite tricky. So we didn't invest a lot into that because we usually don't work with these silicon nanoparticles. So I think if there is a way, if you find a way to immobilize them somehow, then of course you can use EVAnalyzer for it. But I don't know so much about these nanoparticles to say how they can be immobilized, or in our hands. I mean, it worked, but we saw a lot of clusters.
I think
So it
really
sounds
depends
like,
also on the surface charge, and and then for those you might need different coatings. So I think that, yeah, just as we did for the EVs, I think also for other types of nanoparticles, I would start to screen different surfaces.
Yeah. So broadly applicable, but maybe need some modifications of the surface chemistry. Good, good. All right. Well, we're going to end it here. There were some other comments. And I apologize to Leandro, Mohammed, Cora, some others who had nice comments there. But I will share the entire chat box here with Melanie and Nicole. And they, if there's some follow-up, they're welcome to do that. And of course, everybody is welcome to reach out to the authors with any questions. I know that Joachim mentioned that there's a GitHub there where if you have suggestions or if you have specific requests, you can reach out through that mechanism as well. So it's really, really nice. So thanks everybody for joining today and for all of the good discussion points, all the great comments. And I also especially want to thank Melanie, Nicole, and your entire team for this exciting work that I believe is very, very valuable to the EV community, and it's going to bring single EV analysis more into the range of what everybody can do. So thanks so much for that, and all the best on your continuing work. Thanks everybody. Have a good rest of the week, and look forward to seeing you again soon.
Thanks everybody. Thanks, Ken.
Thank
Ciao.
you. Bye.
Thanks, bye.